Good afternoon, anyone. Am I audible?
You are indeed. We can hear you loud and clear.
Thank you, sir. Welcome, sir. Good afternoon, all. I welcome you all to a webinar on drug discovery and clinical trial, which is organized by the Association of Pharmaceutical Research and Scientific Partners, BioLeagues Worldwide, and Research Partners United Innovators on the occasion of birth of Dr. William Procter Jr, sir, who is also known as the father of pharmacy. I would like to introduce about the Association of Pharmaceutical Research. Association of Pharmaceutical Research is one of the world's leading professional association caring for people. APR is a non-profitable professional association meant for research and development in the field of pharmaceutical science and technology. APR is an international forum for researchers, academicians, doctors, practitioners for sharing knowledge and innovation in the field of pharmaceutical science and technology. APR aims to bring together worldwide researchers and professionals, encourage intellectual development, and providing opportunities for networking and collaboration. APR meets with its objectives through academic, networking, meetings, conferences, projects, research publications, academic awards, and scholarships. APR strives to enrich from its diverse group of advisory members. APR brings together pharmacologists, life scientists, pharmaceutical practitioners, and research to advance in the application of the pharmaceutical science and technology to the public. It delivers access to its members the industry's most essential technical information by organizing conferences, workshops, annual conventions, meetings, and provide networking opportunities, both locally and globally. Members have the ability to stay current in their chosen profession, connect with peers, and invest in their futures. I would like to welcome our today's speakers, Dr. Asif Ahmed sir from UK, Dr. Palanrajan Vijay Kumar sir from Malaysia, Dr. Murli Yalpu sir from USA. I would like to welcome our first speaker, Professor Dr. Asif Ahmed sir, Executive Chairman from Mirzam Therapeutics, Senior Advisor to VC and President from the University of Southampton, Scientist and Social Catalyst from United Kingdom. I would like to felicitate sir from biography. Professor Ahmed sir is the founder of Mirzam Therapeutics, whose mission is to make preeclampsia history using exclusive and globally granted patent RNA technology to predict, detect, and prevent preeclampsia. Ahmed sir is also the senior advisor to the president of the University of Southampton. He has held professorship for over 20 years at leading research universities, has over 28,500 citations, and has developed his research to find out why women die in childbirth. The Stanford study 2020 ranked him in the top second scientist in the world. Asif Ahmed sir is a renowned scientist who discovered two protective pathways that put the brake on a preeclampsia, a life-threatening condition in pregnancy. Every year, preeclampsia affects 11 million of pregnant women of the 131 million childbirth worldwide. Usually during the second half of the pregnancy, it causes high blood pressure and undersized babies. There are no therapies to prevent it. The only way to cure preeclampsia is to deliver the baby before the due date. I would like to hand over the speech to us. Please share your valuable information up.
Thank you very much, Dia. Very kind of you. All those words are too generous. I'm going to share my screen and then hopefully I can share my slides with you. Let me know if my slides get shared with you. Oops, I don't know what happened. Let me stop sharing. Somehow I did something wrong, obviously. Apologies. Right. It should be now share. Right. Thank you very much, APR, for the kind invitation. Imagine a world free from chronic diseases. We prevent chronic diseases before they happen. We prevent them in utero. We stop the molecular damage caused by preeclampsia and fetal growth restriction. We prevent low birth weight babies and downstream problems. And we do this with one test and with one pill, save the life of both mother and baby. And I'm Asif Ahmed. I'm the founder of Mirzyme Therapeutics and also the founder of Aston Medical School. And I think it's right to talk about the Association of Pharmaceutical Research, which is a non-profit organization. And especially in this forum where it's talking about drug discovery and clinical trials, what I will try to do is to give you the science behind Mirzyme, how we got here, the journey that took us here. But what is the problem of preeclampsia? It affects 1 in 12 women. It leads to high blood pressure and organ damage in the mother and premature babies. It doesn't just affect women during pregnancy. And remember, women make up half the population of the world. It affects them later in life, increasing the risk of premature death from heart attacks, stroke, even dementia. And premature babies have long-term consequences, health problems. And there is no cure for either fetal growth restriction or for preeclampsia. And preeclampsia is on the rise. For a number of reasons. Women have choices. They choose now, and they should, in the 20s to develop their careers. But if you're 35 and older, then your risk of preeclampsia increases more than 4-fold. So that is a trend that is happening anyway, and we need to provide choices for women to make those choices. People are getting fatter, all of us, especially after COVID. And that in itself increases the risk of obesity, increases the risk of preeclampsia. And why chronic diseases? Because preeclampsia increases low birth weight babies. And we know from Barker's hypothesis of fetal origins of adult disease that small birth weight baby increases the risk of chronic disease later in life, such as heart disease, metabolic syndrome. And something like a quarter of such babies are born in the Indian subcontinent. So we need to do something about it. And what's the current management? Is inadequate. Bed rest, blood pressure meds to regulate blood pressure, but doesn't solve preeclampsia. And recently, it's been proposed that in those who are at high risk, low dose aspirin, but there are lots of side effects and it's not really effective in everyone. And there's nothing to prevent organ damage, which is a major risk factor. And in the UK, the NICE guidelines gives a stepwise approach to managing preeclampsia. such as changes to lifestyle, as I've just mentioned, low dose aspirin in high risk pregnancy, monitoring regularly the blood pressure. But when it's uncontrolled, the only option is, as Dia said, we have to deliver the baby. So there's no approved therapeutics available. Myrzyme wants to bring safe, effective, and hospital-free treatment. First-in-class drugs are in development. And the way our approach is to predict with accuracy halfway through pregnancy who will get preeclampsia, and then offer options for targeted therapeutics for preeclampsia or for fetal growth restriction. And so if you think of like pregnancy like a ride in a car, the brakes, the car crashes. This happens in preeclampsia. What we have done is we've developed how the braking system works. And now we are able to put new brake pads on to prevent preeclampsia. So we've now been granted 10 patents in 30 countries, and this has happened after $40 million of non-dilutive research grant. And the team's been together for a long time, and we're sort of 8 to 10 months away from IND studies and 18 months from completing our first diagnostic and therapeutic studies. What is important to make out, I always say this, Myrzyme is not a spin out from a university, it's a spin out from the public purse. And I'll explain that in the next slide. It's because we've been given research grants, which has taken us to this point, from the idea of if 1 in 12 women get preeclampsia, what do the 11 have that protects them? And that's where I want to start the science behind Myrzyme from. So we were the first people back in '97 to put forward the idea in a review that loss of vascular endothelial growth factor may trigger preeclampsia because it may lead to a rise in soluble FLT, the natural antagonist of VEGF, and high soluble FLT may cause preeclampsia. This was a concept which we and others have shown to be case, to be true. What I want to focus on this slide is to show you that if you take placenta from normal and preeclamptic PE cases and expose them to the condition media of that to endothelial cells in culture, what you see is that in the purple, in the normal, there are tube formation, which is a marker of endothelial angiogenesis, whereas in the PE, there was reduction, but doesn't mean it's through any specific. What we did was we then took immunoprecipitated out S-FLT, soluble FLT, the natural antagonist of VEGF, and showed that when you do that, you could restore angiogenic balance. This was the first evidence that a single removal of a protein, that was the culprit protein, could restore balance. And then a whole series of clinical studies were done, which showed that to be the case, that this protein did indeed cause high blood pressure, kidney damage, and it preceded the signs and symptoms of preeclampsia. So if Bradford-Hill causation criteria is that if A is to cause B, A must come before B, and soluble FLT comes before you see the signs of. And so in a way it does this is, back in '97, we had also shown that VEGF stimulates nitric oxide. And so if you have high levels of soluble FLT, it will mop up not only VEGF and PL, placental growth factor, in the circulation, but also forms a dominant negative with the receptor preventing signaling to take place. leading to poor endothelial function. And that may be the underlying cause. But what controls soluble FLT? Because soluble FLT normally in normal pregnancy goes up. So we were looking at if 1 in 12 women get it, what do the 11 have that protects them? That led us to discover 2 pathways that are defective in preeclampsia: hemoxygenase 1 and cystathionine gamma lyase. These 2 pathways produce carbon monoxide and hydrogen sulfide. And what we were able to show that these pathways act as negative regulators of S-FLT. And even before that, why did we think that hemoxygenase was a protective is shown in this slide, where in fact, I did this experiment where we exposed human placental tissues to TNF-alpha and looked at cellular damage by lactate dehydrogenase leakage. And we saw that TNF-alpha caused an increase. And hemin, which is an inducer of hemoxygenase, led to a reduction. But that doesn't mean it's due to hemoxygenase. But in the presence of tin-protoporphyrin, the inhibitor of hemoxygenase, hemin is unable to do that. This was the first confirmation that human placenta had an enzyme, a protective enzyme called hemoxygenase, that protected the tissue from cellular injury. This information, combined with the fact that hemoxygenase may negatively regulate soluble FLT, led us to do these experiments where we showed that if you overexpressed hemoxygenase in endothelial cells, you suppressed soluble FLT production. And if you knocked it out, you increased soluble FLT production. And the reverse was, the same was true for cysteine gamma lyase, the 2 ends. So these 2 pathways act as the braking system. What we did also discover that hemoxygenase was upregulated by statins. And statins are counterindicated in pregnancy. They're category X drug. However, because we proposed a mechanistic approach to this, we were given the approval by the MHRA to carry out a multi-center randomized control trial on statin, pravastatin. And what this showed that although it was safe to be given, it did not suppress soluble FLT and did not prevent preeclampsia. And therefore, we abandoned this approach, but we learned a lot from this experiment about the recruitment, which centers are good for recruiting, which doctors are good for recruiting. Doing the most challenging, world's first randomized control trial on a category X drug gives you a lot of information that is not in the public domain. But what did we learn from all this? We've learned that if you do that blood pressure drugs don't work, right? Aspirin is not effective to prevent preeclampsia, nor are statins. What does work is plasmapheresis. Plasmapheresis has a lot of drawbacks. It does not prevent preeclampsia, right? What it does, it reduces culprit protein, and in a small study, improve patient outcome. But it's highly invasive, requires local anesthetic. Patients have to be hospitalized. It takes 2 to 4 hours to do the procedure, and it has to be repeated every few days of 3-4 days. It's like renal dialysis, needs constant medical monitoring, very costly, can't be something used globally effectively around the world everywhere. And so we wanted to build a drug that is, oops, what's happened here? So a pill. So one test, as I said, you could detect and a pill to it be effective. And also, having gone through the pandemic, we know that vaccines work. A shot that could be how, and I'll explain that a little bit in the sense of how you deliver it. So delivery systems of delivering something that could directly inhibit soluble fluid. So if you think of what I've basically told you so far is if you have increased ROS, low hemoxygenase pathway, a defective hydrogen sulfide pathway, these lead to rise in soluble fluid, and that leads to endothelial dysfunction. So stressed endothelium, stress syncytium, the placenta. And if you could rectify it with a hydrogen sulfide donor or statins, we've already shown it is not ideal. But what you really need is targeted therapeutics. And that took us to look at this while these studies were going on, is to develop in vivo models that could predict with certainty what's going on. And we developed two models, a modified model in the mouse where transgenic mice models could be given soluble fluid and looked at outcome measures, and a surgical model where we ligated the ovarian arteries and were able to do that again in the mouse. It's been done in the rat, but not as effectively as we've done so in the mice model. And what this did is we were able to show that in our mouse RUP model, blood pressure went up and our treatment brought it down. Same was true for the kidney improved, vasculature improved, and maternal soluble fluid levels went down. And there was an increase in live birth. So both in this surgical model, we saw a shift from preeclamptic symptoms to normalization of symptoms. And you can see this in this picture of the pups, how treatment improved and how the placenta becomes vascularized again, which is avascular before treatment. And again, in the transgenic mouse model with low soluble fluid, low hemoxygenase, high soluble fluid, we saw the same kind of behavior of our drug. And in the drugs that were to look at fetal growth restriction, we saw a shift, 10 centile in the curve at the top, you see, is where the fetal growth restriction, and you can see where the patient group is. And when after treatment, it shifts to the right as the weights improve. And you can see fetal loss is significantly reduced. And fetal placental ratio is a health indicator. And so we saw a balance moving towards a more healthy after treatment. And these have led us to get the patterns on what I've just been talking to you about. And again, here you see the vasculature in the placenta shifts more to the normal after treatment with the drug. So in a way, most people have focused on the downstream events. We've focused on the upstream events and identified two pathways that are defective, which act as a brake on preeclampsia. And this, when the brakes are defective, soluble fluid levels go up. And then we ask the question, how do these brakes regulate S-fluid? And that's another whole story which involves microRNAs, and we've identified microRNAs that directly target the third prime end of S-fluid, and that's is a whole new area that is exciting and improving, which we believe in the light of the last 2.5 years of going through vaccinations approach with AstraZeneca vaccine and the Pfizer vaccine, those two delivery system offer us a huge potential to prevent preeclampsia. And I want to end by to tell you nothing to do with this, but about 2004, I used to be a professor in the University of Birmingham, and I met with Richard Ernst, who got his Nobel Prize for NMR spectroscopy. And Richard and I were having tea, and Richard says to me, how's your poetry going? And I said, I don't have time for poetry. And he gave me this cartoon, which I've modified. But the two P's of this are his, and the other three are mine. And what Richard said, in our profession, you need an additional passion if you want to dream about Stockholm and I said to him, Richard, you can say that because you've got the Nobel Prize. If I said that, people think it's ridiculous. But what I've learned is that you need purpose, because what he was saying, no, you need it to be creative. But what I've learned is that you need purpose, because when you have purpose, you have focus. When you have focus, work isn't work. As Steve Jobs once said, the vision pulls you. But then you need persistence, right? Persistence is where most people fail. They give up before they see the Holy Grail. So you need persistence, but even with persistence, you will hit a brick wall where you can't go through and you can't go around. And that's when you need persuasion, when others come to your view and it's no longer your vision, it's a common vision. That's where you need the power of persuasion. And so this is what I would say to young people there. Don't go for a small, quick fix. Take the long road. And when you do that, then a movement starts. And Let's Make Preeclampsia History came about because my student, when she finished her PhD, during lockdown, came to my house with a present and a card. When I opened the card, it said, "Let's make preeclampsia history." So please join the movement. Let's make preeclampsia history. Thank you. Oops. Happy to take any questions.
Thank you so much, Shah, for sharing your wonderful information with us. Any questions from the audience, please? Thank you so much, sir.
Pleasure.
I would like to welcome our next speaker, Dr. Palanrajan Sir, Vijay Kumar Sir, Head of the Department of Pharmaceutical Technology, UCSI University, Faculty of Pharmaceutical Science, Kuala Lumpur, Malaysia. Dr. Palanrajan Vijay Sir Kumar received National Doctoral Fellowship under Quality Improvement Program sponsored by All India Council of Technical Education, New Delhi, India. He completed his PhD in Dr. Hari Singh Gaur University, a central university in India, formerly known as Sagar University, from November 2007 as a full-time research scholar. He published many research findings in high-impact journals, among that one of our original article titled Dendrimers in Oncology, An Expanding Horizon, was published in ACS Publishers Chemical Reviews. He has undergone various training programs to update knowledge and skill, as well as update recent developments in the field by spending time to read research, review article, as well as international patent. I successfully developed course modules for BSc Formulation Science in hybrid mode, MSc Pharmaceutical Technology mixed mode, and MSc in research mode, and now involved in developing Diploma in Cosmetic Science and Diploma in Packaging Science along with my team members. I would like to, sorry, sir.
Okay. Um, Thank you, Ms. Divya, and thank you for APR for invitation. Is it I am audible to everyone? Hello.
We can hear you.
Extremely sorry, sir. Yes, sir, you're audible.
Okay. Thank you very much.
Like to,
Okay. Is it my slides are visible to everyone?
yes, sir, your slides are visible.
Okay. Thank you very much, Ms. Divya. So today's my topic was revolution in the synthesis of gold nanoparticle. Actually, the gold nanoparticles are become famous during this COVID-19 pandemic because it is used in COVID self-detection kits. And these gold nanoparticles having a unique property compared with some other nanoparticles. It have the surface plasmonic resonance effect. Also, it have the high surface area to volume ratio, and it capable to conjugate with the various ligands and drugs, as well as the peptides, polypeptides, like anything we can put it coat on the surface of the gold nanoparticles. These gold nanoparticles mainly, initially, it is used in the biosensors. Uh, After that, it is used in the photothermal agent for cancer cell treatments. Then it used in the surface enhanced the Raman spectroscopy, and we can detect even a single molecule by using a Raman spectroscopy because it increased the signals, output signals of the drugs in the Raman spectroscopy up to 10 to the power 10 times higher. It is used in the tissue engineering and regenerative medicines and optical imaging, so on. And the unique property of this gold nanoparticle, we can produce this gold nanoparticle in different shapes. So for nearly 9 shapes of the gold nanoparticles are successfully produced. The first shape is usually the spherical shape. It is widely applicable in biosensors as well as in the drug delivery systems. Thereafter, the branched leg structures, road shape, cubical shape, triangular shape, uh, bell flower shapes, quasi ellipsoidal shape, hexagonal shapes also are produced by various researchers. These gold nanoparticles are produced successfully by the scientist called Dr. Terwich. So the method itself called as Terwich method. In that he was used the hauric acid, the presence of reducing agent sodium citrate at 100 degrees centigrade, he capable to produce the gold nanoparticles. It is a very simple method and anyone can produce in the huge volume also. This whole idea is came from Michael Faraday, but the successful synthesis of the gold nanoparticle is from Terwich method. After that, there are a lot of modification happened in this method. Nowadays, in the pharmaceutical sector, we are using the burst synflip method. And also recently, we noticed that there is a lot of changes happened in the gold nanoparticle synthesis. Green synthesis, seed growing methods have become famous among them. Usually, the gold nanoparticles are prepared in the large scale by top-down approach or from the bottom-up approach. Nowadays, mainly we are using a bottom-up approach. The top-down approach is the bulk gold metal is converted in the form of the gold nanoparticles. Bottom-up method, the gold ions are aggregated to form the gold nanoparticles. The recent techniques capable to form the unique size gold nanoparticles through the bottom-up method. Usually these gold nanoparticles are synthesized by using three types of the chemicals. One is the metallic precursor and reducing agent and the capping agents. These three agents combine together to form the gold nanoparticles. As per the Terwich method, we have to use 100 degrees centigrade, but the issue is if we are using a monoclonal antibodies or antigens like materials, we want to load it in the gold nanoparticles, the temperature is not suitable. The recent modification, we are using a extra reducing agent to nullify this temperature effect to form the gold nanoparticles. Mainly, we are using a sodium borate, ascorbic acid, sodium ascorbate. Also, we are using a capping agents to hold the antigens, antibodies in the surface of the gold nanoparticles. Here, we can eliminate the high temperature to synthesize the gold nanoparticles. Bust-Stringhoff method, very famous method, because we know that many of our drugs are lipophilic in nature, but the gold nanoparticles are previously prepared in the aqueous solution. But in this method, you can see that here we are using a mixture of the solvent toluene plus dodecanethiol plus water and auric acid. So it forms the two layer. After that formation of the two layer, we can dissolve the drug in the toluene and dodecanethiol mixture. Then if we add the tetraoctyl ammonium chloride and followed by the sodium borohydrate, it forms the gold nanoparticles loaded with the drug. So this is the advantage of this method. To load the highly lipophilic drugs in the surface of the gold nanoparticles. Nowadays, we can form the gold nanoparticles without using reducing agents, and also without using heat, also by using the UV spectroscopy or UV lights. Just you pass the UV light in the auric acid and sodium citrate mixture, it forms the gold nanoparticle within few hours. And the electrochemical synthesis. In the electrochemical synthesis also, we can avoid the sodium citrate also. Here the auric acid is distributed in between the anode and cathode and pass the current. At the time, the gold ions will settle in the surface of the electrodes. After that, shut down the current, and once you get the proper size nanoparticles, it can be scrubbed out to precipitate inside. One more method is the template synthesized method. In the template synthesized method, the templates are used to load the auric acid, thereafter add the reducing agent sodium citrate, and here we can produce the required shape of the nanoparticle. The first method, they produce the rod shape gold nanoparticles. And now the laser ablation synthesis of the gold nanoparticles are famous. Here we can use light 532 nanometer or 1064 nanometer light. It is in the visible region. It is also called as the green method. In this method, the gold plate was irradiated with these lights, and it capable to form the gold nanoparticles. The sputter coating is commonly used in the scanning electron microscope and other microscopic technique. Here we are producing the gold nanoparticles by flashing the argon plasma in the bulk metal. At the time, it forms the gold nanoparticles. These nanoparticles can be coated in the surface of further any material you want to enlarge up to 100,000 times. We can enlarge by using a scanning electron microscope. We can visualize this one. The green synthesis now become very famous. There are a lot of well-characterized plant extracts are available in the market. These are having a reducing activity, or we can use the enzymes or microorganisms to produce the gold nanoparticles. These are the very successful method at this moment. But gold nanoparticles are having some disadvantages also. So there are a lot of research is going on to produce a gold nano alloy to hold the more amount of the drug and improve the activity of the drugs more than 10 to the power 5 times, 10 to the power 10 times. So usually these gold nano alloys are synthesized by chemical synthesis method or green seed method. Here it is also looks simple, and we can produce this gold nanoparticle in very large quantity. See, usually the gold nanoparticles are produced by using the auric acid and adding the reducing agent and stabilizing agent. During the production time, we can add one more metal ion, and then heating and stirring, we can produce the gold nano alloy. Here we can get the matrix device. That means the gold will be mixed with the other metal alloys uniformly, like that matrix device are formed. But nowadays the research was going on. First they will produce the gold nanoparticle. After that, the other metal ions will be added in the surface of the gold nanoparticle. That is also possible. So first we can produce the gold nanoparticles by using a conventional method, just auric acid, and add the reducing agent and stabilizing agent. And if you need any drugs also can be added here. At the time it forms the gold nanoparticles. Once the gold nanoparticles form, after that other metallic precursor can be added, and you can add the stabilizing agent, and the reducing agent will be selected based on the other metallic precursor, and it forms the other metal coated gold nanoparticle. So we can see the gold nanoparticle further coated with the other metal. So this is also possible. How we can avoid the heating of the synthetic methods by using some unique chemicals. Here you can see the citrate trimethyl ammonium bromide. This citrate trimethyl ammonium bromide can be used in the preparation of the gold nanoparticles in the presence of sodium borohydrate. If we mix all these things together, the gold nanoparticles can be produced in very low temperature itself, 0 degree to 4 degree temperature itself. This method is mainly useful to load the siRNA, mRNA, DNA-like materials in the surface of the gold nanoparticles. And also we can produce the gold nanoparticles in the presence of the silver ion to make the silver gold alloy nanoparticles. It have some other unique properties, but usually this method, the gold nanoparticles are produced in spherical shape, but in the presence of silver nitrate, if we make the gold silver nanoalloy, it is usually in the rod shape. The whole presentation is based on our recent publication in the current pharmaceutical design. Based on this basic idea, we have recently synthesized the new gold nano composite. Here we used the anti-cancer drug tamoxifen and made it as an inclusive compound by using a beta-cyclodextrin. After this formation of the inclusive compound, we loaded in the surface of the gold nanoparticles in the presence of ketosane. The advantage of this gold nanoparticle is it have the plasmonic resonance activity. It's usually happen in the surface of the dielectric metal surface in the presence of the light. The electrons will be activated and it capable to enhance the activity of the drug very well. So this plasmonic effect of the gold nanoparticles can be achieved in around 530 nanometer. This is the advantage of these methods. And also you can see this gold nanoparticle, even though we produce in the lab with very simple procedure, it have a very unique shape. Each and every molecules we can produce in the spherical shape. At the same time, we can coat the drug in the surface of the metal and nanocomposite. The presence of chitosan, it holds the drug very well in the surface of the gold nanoparticles, and also it will taken up by the cells. For checking the activity of the drugs, we have conducted the excelligent technique. In that we are used the RTCA instrument. Here we can see it. Usually the CACO2 cells will grow very fast within 2 to 3 days. But if you are treating with the drug, it kills the CACO2 cells. At the same time, if you load the small amount of the drug, even you can reduce 100 times the amount of the drug, and you load it in the gold nanoparticle and you treat with these cells, and the cells will be immediately dying. At the same time, these gold nanoparticles presence of the little amount of the drug, not toxic for the normal cells. How it is happening actually, once we incorporated the gold nanoparticles in the cacao2 cells, we can see the time increase, the absorption of the gold nanoparticles in the surface of the cells will take place initially. Thereafter, it goes and invade inside the cells. That can be easily visualized. Inside the cells, the gold nanoparticles are invaded through the fluorescent microscopy, and also we can see the emissions fluorescent spectra of the gold nanoparticles through this fluorescent spectrum. Because of that concept, we can use the little amount of the drug to enhance the activity. At the same time, once the drug was entered the inside, we can pass some visible light. Then we can activate the drug very well to kill that particular cells. Okay, this whole work was we have recently published in Turkish Journal of Pharmaceutical Sciences. So this simple research work is funded by Fundamental Research Grant through Ministry of Higher Education, Malaysia, as well as from UCSI University. These are our references. If you have any questions, please, you can email or you can ask it now. Okay.
Thank you so much, sir, for the wonderful information. I'm sorry for the inconvenience, sir.
Okay, no problem.
Any questions from the audience? Thank you so much, sir.
Okay, thank you very much.
I would like to welcome our next speaker, Dr. Murli Yalapu, sir, Associated Professor from Department of Immunology and Microbiology, member of South Texas Center of Excellence in Cancer Research at the School of Medicines in University of Texas, Rio Grande Valley, Texas, USA. Dr. Murli Yalapu, sir, is a tenured associate professor of the Immunology and Microbiology Department and a member of South Texas Center of Excellence in Cancer Research at the School of Medicines, University of Texas, Rio Grande Valley, McLean, Texas, USA. He is currently seminar coordinator for DIM STCE CR Seminar Series. His current research primarily focused on the development of nanomaterial for improved therapeutic potentials of clinical drugs using nanotechnology. Additionally, through Cancer Immunology Institute, his laboratory will generate safe and effective nanoformulation for cancer immunotherapy. He is also world top second in highly cited scientist. He served as editorial board member, associated editor, and senior editor roles for various journals. He has published over 160 peer-reviewed articles and reviews in journals, 12 book chapters, and over 125 conference abstracts, and now over 25 invited talks. He worked for cited over 12,430 times with an H index of 55 and I-10 index of 128. I would like to invite sir to share the wonderful knowledge with us. Welcome, sir.
Thank you, everyone, and good evening who are in the India, and good afternoon, who are in the other places, and good morning who are in the United States. Can you hear me?
Yes, sir, we can hear you.
Yeah, thank you. Thank you.
Sir.
So I'm going to talk about a new concept called nanomedicine. It's not really new, but to some of you, it may be new. So nanomedicine discovery to cancer treatment. So I have divided this topic into two items. One would be introducing nanotechnology or nanomedicine, and next, how we can use those into a cancer treatment regimen or modules. Before I go in detail, I want to just introduce my lab focus. And here is a holistic approach we take right from the prevention aspect, imaging, diagnosis, and theranostic aspect of the cancer. And also we are also interested in developing some of the strip-based rapid test methods for COVID and other diseases, including cancer. And we are also introducing some of the new concept called nano immunoimaging and also nanotherapy and immunoimaging. So without further delay, I want to just introduce section 1, that is cancer and how we are going to introduce a nanomedicine into cancer. So if you see the statistics 2021 of the United States, close to 1.9 million people are affected by this disease, and also over 600,000 people deaths occurred each and every year because of this disease. So identifying or improving the therapies that can tackle cancer is highly at most unmet clinical need. So if you see the cancer management, you can come across like either preventive modules or the screening protocols or diagnosis and treatment. So if you can find anything that can fit in these 4 major areas that can help the patient compliance or improvement in the therapy, or maybe you can minimize the side effects. For example, if you use any nano-based material that can detect early, that can be used as a screening or diagnostic agent in the cancer treatment or cancer management. And if you can find out early nano material that embedded with some kind of preventive agent, that can be used in the preventive regimen. So no doubt in the treatment, there are so many agents that is embedded with the nanoparticles or nanomaterial that can be highly useful in the treatment modality. So just to introduce, although my previous speakers, they mentioned, what is nanoparticle and how it can encapsulate, therapy or drug agent, or maybe any imaging or contrast agent. So if you see nanomaterial, not only in the clinical era, but also you can see the use of these nanomaterials in the construction and also in the electronics and also some of the diagnostic tests that we can use. So it's a combination of a wide variety of studies that can applied for the nanomaterials that utilizes nano science or material science or biology that can be implemented, physics, chemistry, and various other elements that are involved. By combining all these efforts, that nanomaterials can be applied in the therapy, separation, diagnosis, and so on. You can see on the left bottom of this screen that nanomaterial can be used for imaging, biosensing, cell sorting, and proteomics, hypodermia, drug delivery, and so on. So one can imagine that what are nanoparticles, how one can visualize, or one can imagine that if you see this slide, it can give you holistic approach or a brief information that how if you look about a glucose molecule, we consume a sugar every day, but if you see the structure of the glucose, so if you measure the size of the glucose molecule, it's a 10 power minus 1 nanometer in size. If you take any proteins, those were close to 1 to 5 nanometer in range. And if you DNA and viruses, those were like 10 to 100 nanometer in size range. So whatever we talk, mostly we will be talking today from 1 nanometer to 100 nanometer more precisely. But in general, we'll be talking about 1 to 1000 nanometer, that is nothing but 1 micron in size. So if you see this in the size range 1 to 100 nanometer, at the bottom of this slide, you can see wide variety of structure that those were included, those nothing but various type of material that used to make nanomaterials. So that can be polymeric micelles, liposomal nanoparticles, and dendrimeric nanoparticles, gold nanorods or nanoshells, quantum dots, polymeric nanoparticles, and so on. So what are the issues that are exist in the delivery area? That you can see the listed on the top left-handed side, like it includes low solubility, and it has a high systemic toxicity, low bioavailability. They have some PKPD, nonlinearity, and low targeted efficiency. And irrespective of all these, you may come across, like even if you use the same drug molecule again and again, the cells develop resistance. To overcome all these, the nanoparticles may have some better therapeutic effects that we are going to talk more in detail. So again, here the slide giving you some other introductory information that if you divide into different particle size range, so 1 to 10 nanometer, that you can use dendritic nanoparticles or gold nanoparticles or magnetic nanoparticles or carbon nanotubes. And when you divide into 10 to 200 nanometer, there were other nanoparticles, with respect to liposomal pharmaceutical nanoparticles or polymeric micelles. So this will give you, like, visualization purpose, like, you can imagine that, different particles, different size range, different constructs can be made that can be used for polymeric nanoparticle based or any other nanoparticle based, but that can be applied for both therapy and imaging aspects. And what are the issues commonly exist with the nanoparticles? The primary basic issue would be a stability of these nanoparticles. Whenever we make a nanoparticle formulation, all the nanoparticles, although they are smaller in size, due to van der Waals forces, or they come closer together and then they form a aggregative formulation that may not be dispersible in water or may not be dispersible in any other solvents. So therefore, the prime motto of whenever we make any formulation, the formulation should be very stable in either in the solid state or in the liquid state. So therefore, we must be using some of the stabilizer that can keep apart one nanoparticle. Then we need to also consider, like, once you encapsulate any imaging or drug agent into this nanoparticle, what would be the release profile of these nanoparticles? So that must be known, that you can see that the nanoparticle either can be degraded and release your therapeutic payloads, or maybe slowly diffuse from these nanoparticle constructs. And also you can see on the right-hand side, various other modules or exist that can be improve the release, or maybe it can burst release based on the, if you use any specific external stimuli, okay, such as temperature, pH sensitivity, redox sensitivity, and so on. And what another concept that you commonly experience whenever you use nanoparticle, particularly in cancer therapeutics, there are two items here is listed with red color, that is passive targeting and active targeting. The passive targeting is nothing but the nanoparticle, some specific nanoparticle size, they can go via leaky vasculature into the tumors, and then they locked up into the tumors. So therefore, these nanoparticles will reside into the tumor vasculature and at the tumor side, so that the therapeutic payload will be released over the period of time, so that you may intend to get desired achievable therapeutic benefits with the nano formulations. Whereas on the other hand, the active targeting is nothing but you have a nanoparticle that is encapsulated with any drug or therapeutic moiety, but also surface decorated with any other cancer targeting by using any ligand or antibody or aptamer and so on. So that these nanoparticles, when they go through via vasculature, and they can specifically bind to the cancer cells, not on the normal cells within the tumors. And you all need to remember that the tumors are not just with the tumor cells. It composed of various other cells such as macrophages, and so on, like the fibroblast, you name, like, there have a wide variety of cell population that exist within the tumors. So therefore, active targeting would be much beneficial so that they specifically bind to those cells and then give these therapeutic load into only to those cancer cells. So what other that we need to know before we apply any nanomaterial, that is nothing but we need to know the architecture of these nanoparticles. That includes not only the particle size, but its shape of these nanoparticles and what targeting might be that we have on the nanoparticles so that we can explore for the targeted way that we can deliver things. And also what is the surface decoration that we have, either it has any positively charged, the surface modification is benefiting, or negatively charged or neutral charge, or either any, it could be hydrophobicity or hydrophilicity. So with all these, once you make nanoparticles, if you see this slide, it will give you some information that if it has small or very small in size, like 1 nanometer, whenever you inject these nanoparticles, these will be cleared by some of the organs within the body. And if you have a positively zeta potential, that will be toxic to the cell. And where you have a particle size, perfect particle size from 10 nanometer to 200 nanometer, those obey the EPR mechanism. That would be the size range that we need for effective delivery to the tumor cells. This is another slide. It's a very busy slide but simply what I want to tell from this slide is that you modify nanoparticles, or once you make the nanoparticle based on its physical properties, size, charge, and so on, and what is the composition that is involved, whether it is a liposomal based, dendritic, whatsoever, then what targeting might you have on the nanoparticle, that would determine what we were going to see. Once you have all this modification, it could be improving the circulation of the nanoparticle in the blood, or it could be improving the specific tumor delivery, not on the other major organs, or maybe it is locking up into the tumor via EPR mechanism or targeted way. And also whether we can introduce some of these stimuli delivery of these nanomaterials. This slide is giving us basic information when the nanotechnology started. It's not started just a few years ago. So if you see that left-hand side, way earlier in 1964, liposomal structure was published. So the liposomers were the first nanoparticle formulation that was applied for delivery of therapeutic agents, for example, doxorubicin. And if you keep coming, and then you will see a latest nanoparticle formulations, cell-cloaked nanoparticles, and so on. But in this scenario, coming the journey from 1960 to 2015, you may come across wide variety of technology. Nowadays, you can also see everybody might be using in the clinic to deliver the COVID vaccines, that nothing but using liposomal formulation. So whenever you see, compare with a few of the nanomaterials that are widely exist, the liposomal formulation obey all type of physicochemical and biological aspect, delivery aspect, physiology aspect, and then designing a nanomaterial aspect. This all will be applied for the liposomal formulation. You can also see some of the other novel methodologies exists, but there may be some gap that need to be filled with those nanomaterial to be able to obey all these aspects in terms of delivery. Now we will go to section 2, that where we will be talking about if you see any nanoparticle that are approved by FDA, either for the delivery or treatment, either it could be for theranostic or imaging application, it has been divided from over the years, that color bars representing various type of material that has been approved by FDA for various indications. And also the C and D is giving us more information still, that number of clinical trials that are undergoing with the various types of nanomaterial that could be protein-based, polymeric-based, polymeric micelles-based, liposomal or metallic, and other nanoparticle-based. As I mentioned before, 1967 or something, I mentioned earlier, what are the nanomaterial that widely used? That was liposomal formulation. Before that, there was a formulation called iron oxide-based nanoparticle formulation that used to use for iron deficiency purpose. So you can inject this nanomaterial so that you can be getting more iron through this iron oxide-based nanoparticles, because it really slowly iron with this nanoparticle. In addition to that, iron oxide formulation used for MRI applications, that is called Faradex IV earlier, and there are newer formulations that are approved by FDA currently for various image MRI contrast reagent purpose. And these are all we never use. Some people might have not seen or come across these various type of nanoparticle formulation. But I want to give you one best example that you all might be using this medicine that is nothing but like microparticle based Bayer formulation that is coated with the aspirin. So aspirin, it's a well-known drug molecule that we used for various indications, headache, or maybe that can be as a preventive agent for cardiac associated issues. So earlier we used to use this aspirin in various forms. So nowadays you can get these into a nano or microparticle base that can be releasing more quickly the same therapeutic load. And here are some of the examples of FDA approved nanomedicines that could be obeying EPR effect, that is nothing but just nanoparticle that is recruiting into the tumor by EPR mechanism. So that is Ablation or any other nanoparticles, Faradex IV, or other paclitaxel-based nanoparticles, it could be a polymeric micelle base. And lately, there is much focus on developing targeted nanoparticle-based formulations that coated with a wide variety of antibody or some other aptamer or fragmented antibodies so that it can be delivered more precisely to the tumors. So what is the major issue with the doxorubicin that we want to make nanoparticle formulation? It binds to the DNA and it breaks the DNA so that it can kill the cell. At the same time, this doxorubicin molecule toxic to the cardiomyocytes also. So to minimize this, nanoparticle formulation was must. Therefore, the doxil formulation was created. Okay, it's based on liposomal formulation. What it does here, you can clearly see the cardiotoxicity is completely reduced or it may be absent. So that's a major milestone that we want to achieve with this particular drug. And if you see later on, the same technology can be applied for various other drugs. Here I've been showing you other antibacterial or antifungal agent. The same technology can be used. Another best example is a paclitaxel anti-cancer drug So how that it works, it stabilizes the tubulin polymerization. So it doesn't discriminate normal cells versus cancer cells. So therefore, what happened here, we develop a formulation that is like, FDA approved formulation. Again here, it's nothing but human serum albumin decorated or bound paclitaxel nanoparticle formulation. So if we talk about targeting, it obeys both either passive and also active targeting. So it can bind to the GP60 receptors that exist on the various type of cancer cells, but primarily it is widely used for breast cancer and lung cancer so far. And also it can be used for the pancreatic cancer cases also. The same technology are being implemented or under the clinical trials for various other cancer indications too. So you see the fall and rise of these plain paclitaxel formulations, that is Taxol, and once Abraxane was created, then the use of Abraxane has been increased quite a lot, and then downfall of this Taxol formulation. So there are other depots that we can also apply. You can make the nanoparticle-based depots, and then you can ask for the sustained and long-term delivery with the nanoparticle-based formulations. And as I mentioned before, iron oxide-based formulation, still exist for various implementations here, iron oxide deficiency, and also for imaging purposes. Although it's iron oxide based, it doesn't interfere with the other contrast imagers like X-ray, CT, and PET, and SPECT, and so on. Additionally, magnetic nanoparticles can also apply for hyperthermia application that can be also used for cancer treatment, particularly in prostate cancer and brain tumors purposes. So here you inject right into the tumors into the brain, and then you may expect the cooking these tumors with the hyperthermia so that you may experience better therapeutic benefits with this kind of therapy. And what we usually learn from all these technology or approved formulation, still there are some issues exist. For example, when you talk about Faradex formulation, it has been used for about over a period of a decade, but there was an issue with the stability. Therefore, this formulation was recalled, and therefore you may expect to see more and more novel formulations that are either FDA approved or still under investigation, but those are all based on iron oxide based, because those are like highly biocompatible and biodegradable. And when you see the bind 014 nanoparticle formulation, you might recall this. This formulation was made by the same group what mRNA vaccine nanomaterials made. But this formulation didn't go through very well because of a lot more chemistry, or maybe it didn't improve the delivery of the therapeutic payload, in this case, docetaxel. So same way, like, too much of chemistry is not always helpful, but, you may construct, very fascinated nanomaterial. We think that, that can improve the delivery aspect, but it's not the case in many examples here. One of the example is polymeric micelles. It did go to phase 1 and 2 level, but it didn't pass through the phase 3 level. Considering all this, what we thought that, we want to create something very meaningful, and then that can be applied for various therapeutic applications. Here we come across the various methodologies. We implemented those, and then we make a nanoparticle formulation. Again here, iron oxide based, but we coated with various layers that can be encapsulate or imaging agent or therapeutic agent, but also we put some of the nanomaterial coated with some of the antibody or aptamer that can be specifically targeting to the tumor cells. So here is the data, actual data that I'm sharing with you, that a PSMA targeted nanoparticle that is based on PLTA. So you see right, left-hand side bottom of this, once we make this antibody tagged nanoparticle that is against the PSMA protein that is widely expressed on the prostate cancer cells. So here you see PSMA positive cells, you can specifically localization on the cell surface of nanoparticles. That indicates these nanoparticles are specifically binding to those particular cells. When you take a PSMA negative cell, there is no nanoparticles that are accumulated on the tumor cell surface. The same thing was, excuse me, same thing was confirmed by cytotoxicity assay with the C42, that is PSMA positive cell. The same concept was proved in the animals also, like when you have a targeted nanoparticle formulation and the last mouse in each stages that you can see the specific accumulation of these nanoparticles at the tumor site. When we excite these tumors, you can see a specific localization of these formulation in the tumors. So I don't want to explain the same, but here, what we did, instead of using any therapeutic payload, so in earlier case, we used docetaxel. Here we use, instead of using docetaxel, we used a small microRNA that is microRNA 205. So 205, it's a tumor suppressor. When you have this 205 in the cancer cell, it's not going to act as a tumor cells. Okay, so it can minimize those tumor activity when you have this microRNA 205. So that means delivery of these microRNA 205 would be beneficial. That's what we are showing here by various physicochemical, biological assay and immuno assays that it's confirming that once you put microRNA 205 in the nanoparticle, that is benefiting towards the improving the therapeutic against the cancer, that minimizing the proliferation and so on. Well, so if you put any tag on the nanoparticle, that would be beneficial. But how about you make any nanoparticle that is self-targeting? So for this purpose, what we did, we clogged these nanoparticles with the cellular membranes. Here we used neutrophils, mesenchymal cells, raw cells, and so on. And then we tested, we clogged these cellular membranes on the nanoparticles. Here it's a PVP and TA-based nanoparticles. And we found that one particle formulation, that is neutrophil here on the right extreme right-hand side bottom of this slide, it is telling you PVT nanoparticles that are clogged with the neutrophil membranes, specifically co-localizing or targeting only tumors, but not on any other organs. So this is the best formulation compared to all other 4 or 5 that we investigated. And how about when you talk about combination delivery, whether is that nanoparticle technology is helpful or not? Here we showed one of the examples that we combined gemcitabine and another drug natural agent that is called gambagic acid. So the synergy we see when we apply these 2 drugs against small cell lung cancer. So therefore, like we implemented this self-assembly based approach to deliver these nanoparticles, we experience better therapeutic effects on the A549 tumor genome graph. There are many ways that we can tackle these targeted way delivery of these material. That here another example is targeting the macrophages, and the next example in targeting the bone in the within the body, so that you can treat the bone tumors and so on. So without further ado, just I want to thank everyone who presented here, and this is my group and some of the funding acknowledgements, and thank you so much. And I'm happy to take any questions if you have. And also here is my phone number, and here is my email address. I would be reachable with this email. And if you have questions, or if you want to extend any collaborations or any other, after this, I'll be welcome everyone, and then hopefully to talk with every one of you who are interested in nanotechnology and therapeutic applications.
Thank you so much, sir, for sharing the wonderful information with us. Thank you, sir. Any questions?
Thank you.
Please?
It'd be good to put it in the chat so people can get the information.
Sure, sir. I would like to thank, on the behalf of Association of Pharmaceutical Research, I would like to express my gratitude to all the speakers for their presence and contribution to making this webinar a more informative and interesting session. APR extends our sincere gratitude to our speakers and participants to take out time from their busy schedule and grace the wonderful event. Thank you so much, everyone.